So this is here I am representing the Department of Community Medicine and the Leptospirosis Research Laboratory of the Faculty of Medicine and Life Sciences, Rajarata University of Sri Lanka. Despite all these researchers, as medical professionals, our interest is how can we diagnose this and how can we manage these patients and how can we prevent this. So those are the main questions. I hope that you will get some answers and the questions to be resolved in future researches. So Leptospira was discovered in 1886, as Dr. Nuan has said. But when we go through our literature, in the history, in Sinhala literature, there were some stories regarding red-eyed demon. So that is Ratakshasanya, which has similar clinical features of leptospirosis. So we believe that leptospirosis should be a historical disease in Sri Lanka. So this is the first publication related to leptospirosis, because I'm going to talk about navigation of leptospirosis research, published by Adolf Weil in 1886. So this is the original research paper published by Adolf Weil. And Stimson demonstrated this spirochete in 1907. And this is the global burden. Leptospirosis is the most widespread zoonotic disease in the world, accounts for 1 million cases, 60,000 deaths, and loss of 2.9 million disability-adjusted life years. When it comes to the productivity loss, it was estimated that $29.3 billion international dollars are being lost due to leptospirosis. And what about the situation in Sri Lanka? With regards to the productivity loss, Sri Lanka ranked at the 4th place. So that's why the global disease estimates consider Sri Lanka is a hotspot for leptospirosis. That's why we need to have more leptospirosis research. So if this is a hotspot, what about Sri Lankan situation of research? The first publication related to leptospirosis was published in 1953 by Lip, and the first paper on mortality of leptospirosis was published in 1959 by Professor Rajarata of Colombo Medical Faculty. And what about the Sri Lankan publications related to leptospirosis from 1950s, 1960s, and 1970s? Are the golden era of leptospirosis research, where without much facilities, they have isolated leptospirosis. They describe, if you go through the history, there are Leptospira 0 was known as Leptospira Piyasenam, Leptospira Veerasinghe, Leptospira Jayavira, like that. Those kind of 0s are there. And from 1979 to 2006, only few publications, and after 2007 and 2008 outbreaks, there were so many publications. Now we are having more than 200 publications. So why leptospirosis is important in comparison to dengue? If we compare the mortality based on reported statistics, as mentioned by the previous lecturers, these statistics are wrong. But even according to the reported number of cases, the leptospirosis mortality is 5 times higher than dengue hemorrhagic fever. So as epidemiologists, our prime duty is to estimate the disease burden properly. Then the others, the policymakers, can make out decisions on them. So we did that. This is one of my PhD publication. And so you can see these light color bars are the cases from the surveillance system, and the middle bars are the hospital data. So you can see that is what you write in the BHD in the first page, in the other side of the first page. So that is indoor morbidity and mortality report. So when we compare those two, of course there should be a difference, but there's a huge difference, because according to the surveillance cases, the highest number of cases are reported in 2008, while according to the IMMR, the highest number of cases are reported in 2011. We use some statistical technique called Poisson regression, and we try to come up with kind of adjusted this thing, and we estimated that annual caseload should be something like this, and annual hospital deaths should be around 311. But again, and I'm really happy that this paper is cited in more than 35 international publications, telling that this is the Sri Lankan caseload. But as researchers, we know that again These case estimates are also based on several other assumptions. So now we all are talking about the plasmapheresis, myocarditis, pulmonary hemorrhages. What about the situation in OPT? There are so many patients who are we may be treating them as febrile illnesses, which may be mild cases, but we are not estimating this. So we did particular study in Ratnapura, by that now it's teaching, by that time it was Provincial General Hospital Ratnapura, and during a lepto peak, during August-September period, and we found that 33 cases which were treated as leptospira, some other febrile illness, later we found that we diagnosed them as leptospirosis, and we got one positive culture result as well, which is very rare incidence related to this. So to the notification system, 28.6 cases are missed from the outpatient department during the peak of leptospirosis. Okay, investigations. I'm really happy that Dr. Sachika, my batchmate, my good friend, and my colleague as a pre-intern, so we participated several international leptospirosis lectures as demonstrators, pre-interns. Now both of us are doing lectures related to leptospirosis. She explained everything regarding the different diagnostic techniques, and these are our findings. So when we are comparing the different diagnostic techniques, whole blood qPCR, serum qPCR, urine qPCR, single sample MET, paired MET, and surveillance case definition and culture. When we compare all these things, qPCR is the best one. But again, as she mentioned, the most important thing is timing, right? So even though the qPCR is best, even in the best diagnostic test, we are going to miss 40% of the cases. So either we have to go for combination of tests, or we need to do, maybe our clinical sense may be better than that because our doctors are good, right? So still there's a huge research gap with regards to leptospirosis diagnostics. And this is the third time this slide is appearing. I need to highlight Leptospira, and once the Leptospira enters the body, we call that septicemic phase, where Leptospira are there in the blood. Then we go to the immune phase. So during the leptospiremic phase, we need to catch the Leptospira organism or DNA of that. That's why we perform PCR, and the latter part we have to go for something like antibody detection, either ELISA or MET. So as the diagnostics are not effective, now as I mentioned, now we have to go by our clinical sense, and specifically the epidemiological criteria. When I was a house officer, the one question that I'm asking is whether you work in the paddy field in Anuradhapura. So if somebody is working in the paddy field, we name that as leptospirosis. That is one of the most important criteria. But what happens during an epidemic is, if it is rainy and cultivation season, we tend to overestimate the disease burden. Because I'm talking much about disease burden because our topic is disease burden and estimation. And dry season, we may be underestimating the disease burden. Because of that, in research point of view, the next thing that we can do is going for statistical modeling and statistical prediction of disease. This is what we did, and we predicted, we did some statistical technique called seasonal auto-regressive integrated moving average technique, and we came up with the prediction. You can see these dark color lines are the predicted ones, and light color ones are the observed ones, which are overlapping. So we can use them for prediction of outbreaks specifically that are occurring during non-seasonal times. That is another aspect. So coming back to clinicals, I'm not going to talk about much about the clinical aspects because other speakers are talking about the clinical aspect. But still, when we go through the Sri Lankan publications, almost all the complications have been reported from Sri Lankan patients: renal involvement, liver involvement, cardiac involvement, lung involvement, neurological, pancreas, bleeding, spleen. Even though the publications are not there, there are so many patients who are admitted to the surgical ward with acute abdomen, and later they they are transferred into the medical ward for managing of acalculous cholecystitis, which is happening in Sri Lanka. So almost all are there. Renal complications are the most common complication, while pulmonary hemorrhages is the most deadly complication according to the reported numbers. So why there's a huge clinical diversity related to leptospirosis? Still there's no proper answer, but the most common hypothesis behind this is, now when it comes to dengue, there are only 4 strains. But Leptospira, we have more than 250 serovars, which might give rise to 250 different clinical manifestations. So many different manifestations. So we believe that this might be the reason. And we found that something interesting. So we need culture isolation. We need to isolate, because if we are to develop diagnostics or develop vaccines, we need to identify what are the circulating serovars in Anuradhapura. Of course, in our studies, we found that the Anuradhapura serovars are different from Ratnapura. And some are similar. So we are in trouble. So to describe clinical features, we need culture isolation. And to optimize the diagnostics, we need culture isolation. Of course, for development of vaccines, we need culture isolations. With all these works, our lab received the accreditation by the International Leptospirosis Society with 99% accuracy for mat proficiency testing. So now we are running this lab. So we are providing diagnostics for the MICU as well as the wards right now. But still there's no proper mechanism. We are doing as a part of, while performing the research experiments, we are giving these results to the ward patients as well. And fortunately, we isolated 25 different strains after experiments of 4 years. We have now 25 new strains, which we have named as FMA's KW1. What is FMA's KW1? This Faculty of Medicine and Life Sciences, Karavanalla 1, because we isolated that patient from Karavanalla. Faculty of Medicine and Life Sciences, Karavanalla 2, Avisavella 1, like that. So we have strains which we named from the name of the faculty. So we have 25 isolated. Those are not new species, but new strains with a different genomic sequence. Even though there's a huge disparity, this is an interesting thing. We found that in dry zone, there's a common pattern of circulating serovars. For example, now when it comes to dry zone, Leptospira interrogans and Borquitaceae are the common circulating serovars, while in species, while in wet zone, interrogans and Curtionary are the common. Those are not very specific results because we have only 25 isolates, but we found that there's a pattern, which is a good sign for future research, specifically for prevention or developing the vaccines. This is something that the microbiologists are in love. So this is what is called Dingus disc, the typical feature of Leptospirosis culture. You can see here. So this is Leptospira isolation. So Dr. Dinesha is there. Her MPhil is to isolate Leptospira. She has been trying to isolate Leptospira, and she tried 4 years without any results. So now we have a really good expert with so much of demand from international colleagues. Fortunately, she is not going. The next thing is culture isolation is not enough. We need genomic sequencing. That means if we are to develop a vaccine, we need to find out the specific genomic sequences. But the problem is we have 250 different sequences which might need 250 different vaccines. We don't know if Pratibha is the patient. We don't know what is the serovar that he is getting. So we have to select what is the vaccine. There are human vaccines developed, but still not effective because of this thing. So the aim is to we sequence all these things and to find out whether there are regions which we can target. If there's a common region for at least majority of these zeros, we can make use of that particular range and develop vaccine. So that is what we are trying right now. But unfortunately, we don't have grants right now. But we are trying in whatever the facilities available. So the problem, the discrepancy between developing vaccine and this thing is, as I mentioned, the number of zeros and number of vaccines. We need more vaccines. So that is the problem. The next thing is how we can explain the difficult complications like myocarditis, pulmonary hemorrhages, and other complications. So we did some investigation related to cytokines. So we tested about 22 cytokines in our lab, and initially we didn't find any association, but we performed multivariate analysis and we found that there are some associations. Those are not very specific and accurate results, but we found like interleukin 5 might be associated with lung involvement, interleukin 8 might be associated with hypertension. But we need large experiments to give more solid evidence with regards to this. So clinical management, I'm not going to discuss, but the antibiotics, the problem associated with the antibiotics, of course, we know the antibiotics, but the problem is when we compare with the pathophysiology, septicemic phase is there. So when it comes to immune phase, antibodies are there. So Leptospira are not there in the blood. So what is the use of antibiotics? But still we have to use it. So here in this lecture, I need to mention that I'm not going to suggest any clinical management because those are research findings. But the problem to be solved is the use of antibiotics at the latter part of the disease. So in some cases, it is effective. So we need to change our pathophysiology as well. And the role of steroids, some studies say that it's effective, some studies say that it's not effective. So we need to find out. The plasmapheresis she mentioned. And critical patient management. So what are the options available? I know that there's some interesting thing is going on related to noradrenaline adjusting doses or something like that. I got to know from the medical officers there in MICU. So of course you need to publish those things. So those are very important things, questions to be solved. Prevention, if you are MOH or in public health, prevention is the most important thing. As Dr. Niranjala has highlighted, we have a problem in doxycycline. Antibiotics prophylaxis. So if we need some solid evidence, we are going for systematic review. And one of the best reviews that we are having is Cochrane review. There's a Cochrane review published related to prophylaxis of doxycycline, which says that doxycycline is not effective. But we don't discourage doxycycline. I will explain why. There are individual studies saying that doxycycline is effective, but Cochrane review says that it's not. There are diseases which are considered as diseases among rich, and there are diseases which are considered as diseases among poor. Right? If you think about diabetes, as rich people are getting diabetes, if you need to find out action of metformin or something like that, there are hundreds of researches conducted in various parts of the world related to metformin. But what about the situation regarding doxycycline prophylaxis? As poor people are getting this, even in this particular Cochrane review, there are only 3 quality studies. So that may be the reason why they say that doxycycline is not effective. So as researchers, we can't recommend, but what we believe is you have to continue doxycycline because there's no other option. Please continue doxycycline in the proper doses. So these are our results this is unpublished work. 32% of the people who received doxycycline got leptospirosis, while 36% who did not receive doxycycline developed leptospirosis. And it seems that there's an effect, like 4% effect, but it is not statistically significant. That is the problem. But of course, there are so many biases related to, as mentioned in the previous, how they take it, whether they are taking properly. We need to analyze those things in this research. Even though I say that this is a disease among poor. So now Europeans are getting leptospirosis nowadays. Americans are getting leptospirosis. Of course, it has been there. So now they have formed something called European Leptospirosis Society, and they are funding. So this is a good sign for leptospirosis researchers, and it will be good for the leptospirosis patients as well. So as researchers, when you become, at one point you become crazy, and if doxycycline is not effective, if there are no vaccines, the next might be, I thought whether we can do something like risk mapping, something like whether Tisavara is safe, Basavakulam is not safe, something like that, right? Whether we can do like that. So we collected some environmental sampling, and we went around Sri Lanka and collected soil samples, and we performed metagenomic analysis, and we found that Leptospira are abundant in the soil, but most probably they are non-pathogenic ones. And this is where our faculty is located. We have selected 26 different sites to collect water, and we tested for pathogenic leptospirosis. Only one was positive, which is an agricultural well, where the flow of water can easily come into that. Then we had this hypothesis. I selected 10 different wells around that place and tested, and 5 of the wells were positive. And we haven't had any clue regarding this, and we repeated the same experiment in 2 weeks, another 2 weeks, another 2 weeks, and another 4 weeks, and we found this in first testing, well number 3, 4, 5, 7, 8 were positive, while in the second testing different wells were positive. So it's a different thing. So but there's only one thing regarding this is, when the well is more close to a jungle or a water pool or lake, tendency to get the positive result is high. We believe that those are not research evidence. We hypothesize that it may be, rather than, because we are talking about whether you have worked in paddy field, whether you work in something else, some association should be there with, I think, animal or abundance of animals infected with Leptospira, because Leptospira are there in the proximal convoluted tubules of nephrons, where the antibodies cannot reach there. It's safe environment for Leptospira. So they shed, asymptomatic urinary shedding is there. So that may be the reason. Interestingly, whitewater rafting. What is whitewater rafting? Kitulgal V2. So this one of the explanation that could be related to this is, now a group of people from Mihintale visited Kitulgala, and all the people got Leptospira. And the people who went same day, they didn't get. And in some other day, another group is getting Leptospirosis, while the other group is not getting. That may be an explanation. If that particular time, if the water is contaminated with an animal, they might get Leptospirosis. Those are hypotheses. Those are not solid evidence, and crazy part of the researchers that we are trying to do. So ultimate evidence is we need to do something early. Early referral, early treatment seeking, early treatment, early referral, early management are important with regards to Leptospirosis. So all the research works that we did are not alone by me. There are a group of researchers who are working here. So I thank all of you. So this is the current group that we are having right now, under the great guidance and supports, and the founder of our Leptospirosis Research Laboratory, Professor Janith. I thank you all for this one, and specific for. Still, we have not published at least a single clinical paper. We have published only the clinical database, which is there online. So anybody can use that database, and you can publish your own article using that particular database. We thank you all. So let me show you Leptospira, how we see that.